this meeting. So welcome, everybody. The Office of Programs to Enhance Neuroscience Workforce Diversity within the National Institutes of Neurological Disorders and Stroke is very pleased to welcome you to the Open Stage monthly webinar series. This is a targeted outreach effort for our diverse neuroscience community to learn about NINDS programs and network with NIH and NINDS program staff and the people we work with. So each webinar features a different program, division, or cluster. Today we have some amazing resources and repositories to share with you all. And the goal is really to help us increase the representation of diverse groups in our portfolio. So this month, again, we are excited to get an overview of the NINDS supported repositories for banking and requesting clinical cell lines, biospecimens, and brain bank tissue. Before we get started, I just have a few housekeeping items. If we could go to the next slide. And just a few meeting reminders. All attendees are muted. Please submit questions using the chat function. So we don't have a Q&A box, just use the chat function for all your questions. And the meeting will be transcribed and archived on our website. You can enable closed captioning under the live transcript at the bottom of your screen. And we will send you so much information about the resources shared in this webinar and the recording and everything in a post-event email. So look for an email from us after the event with all of this information. Please do stay connected with us. And with that, I will pass it to Dr. Jane Hettinger.
Hi, everyone. Thank you all. I'd like to join Anna in thanking everyone for coming for today's open stage. Today we'll be hearing from 3 NIH Program Officers, Drs. Christine Swanson-Fischer, Zhang, and Abigail Soyombo, and the CEO and founder of the Braindono Project, Ms. Tish Hevel. They will tell us about the programs they support that provide resources at low to no cost to biomedical researchers, including cell lines, genetic data, human brain tissue, and various other biospecimens. So we'll just go ahead and get started. Our first presenter is a program manager at the National Institute of Neurological Disorders and Stroke, the NINDS. And she manages several really great biorepositories that provide many resources for everyone in the biomedical field. So Dr. Chris Swanson-Fischer, take it away.
Great. Thank you so much, Jane. Appreciate it. So good afternoon to all of you and good morning to some of you who are on the West Coast. My name is Chris Swanson-Fischer, and as Jane mentioned, I'm a program manager, also a program officer at NINDS, and I manage 2 of our large repositories that I'm going to talk about today. One is Biosend, which handles our clinical biospecimens, as well as NHCDR, which handles our human cell and data repository for mostly for source cells as well as iPSCs. You go to the next slide, Jane. First of all, we're going to talk about Biosend, which is our clinical biospecimen repository. Biosend is, you're probably wondering what it stands for. It's the Biospecimen Exchange for Neurological Disorders. It's a U24, which means it's a cooperative agreement. It's not a grant mechanism. And currently this biorepository is housed at Indiana University with the PI, Dr. Tatiana Faroud. It's been in effect since about 2015. So you go to the next slide. And so I think about Biosend's mission as essentially 2-pronged. So really our first goal is to serve as a central repository to bank and maintain clinical biospecimens from a whole host of NINDS funded studies within the NINDS mission. And secondly, it's to distribute these samples from the repositories to approved qualified scientific researchers, both academic and industry worldwide. Next slide. So this is just a current rundown of all of the different diseases or conditions currently in the cell bank. But mind you, we can expand and certainly do so to a whole host of neurological disorders and stroke within the NINDS mission. So currently we have samples for vascular contributions to impaired dementia, as well as other Alzheimer's disease related dementias like Lewy body dementia, Huntington's disease, ME/CFS or chronic fatigue syndrome, Parkinson's disease, and a number of other Parkinsonisms, PSP, spinocerebellar ataxia, and traumatic brain injury. Next slide. So if you're interested in actually accessing some of these existing specimens at the repository, we do so through a pretty standardized approach. These are requests are adjudicated through our NINDS Biospecimen Resource Access Committees, or BRACs. And so we have investigators apply through an online web form. Sort of think of it as a little mini grant application for access to these biospecimens. And so the investigator can work with the repository to determine the cell type, the specimen type, the amount, the volume, and number of subjects they're requesting from, include your biosketch, a research strategy, which I'll happily go into offline for any additional questions, and then a table summary of what samples you're interested in. And then the BRAC convenes on a rolling basis about every other month or so, depending on the nature and number of requests we receive, and they can either approve with revisions, or deny at the current time. And these BRACs or committees, think of them as a little bit of study section members. So they're from the external research community with expertise in clinical research, biospecimen discovery and validation, as well as biostatisticians as well. So currently we have BRACs to support all of our diseases that we're having and maintaining in our repository, including for Parkinson's disease and related dementias, and Parkinsonisms, Huntington's, SCA, ME/CFS or chronic fatigue syndrome, traumatic brain injury, and also frontotemporal dementia as well. Next slide. So you can see there's a whole host of publications. This is just a smattering over the last year or so that I've been using a secondary biospecimen for some of their analysis, including some biomarker and discovery validation, is also supportive some natural history studies. Next slide. And so I think of it as the opportunity for Biosend and NINDS to work with you and to help support the nature of whatever study that it is that you're interested in and working towards. So if you're planning to have a natural history study or a clinical trial, and you're going to be collecting biospecimens prospectively, you may want to reach out to us. We're happy to work with you to support your study, and we can work with you to utilize the Biosend infrastructure for our banking protocols, procedures, and biobanking. Note that the biospecimen collection has to be included as part of a budget of your grant application, and I'm happy to work with you on those details offline. We do want to note that your IRB consent language should include broad support for sharing these biospecimens broadly with academic and industry. If you want to use the existing biospecimens as part of your grant application, we'd ask that you have a letter of support from the BRAC before you submit your grant application that you can include. And then also, as I noted before, your applications are reviewed every couple of months. The great thing is that these biospecimens are available to the scientific community at no cost. So these samples are free to you for running your study. You just have to demonstrate that you have the funding in place to perform those analyses through an existing NIH grant or some other mechanism or institutional funds. Next slide. This is just a quick rundown of some of the numbers. So we have almost 207,000 biospecimens from all of our active studies since the repository was initiated over 7 years ago. Got over 7,000 subjects. You've already seen the disease cohorts that we support from a number of active clinical studies, and over many of them which are multi-site, which is why we have over 122 sites right now. The vast majority of all of our cohorts, I should also mention, have deep longitudinal associated data matched with each. So it's not just you receiving those biospecimens, but you're able to then do a deep additional endophenotyping on those as well. So happy to reach out if you have any questions about that. Again, we've distributed over 33,000 biospecimens worldwide to academia and industry. We've also prepared a number of over 100 quotes requests for NINDS investigators interested in banking with biospecimen, and then over 35 publications. Next slide. So really quickly, I just want to talk about my sister repository that I help manage, NHCDR, the NINDS Human Cell and Data Repository, which has an aims of fourfold. One is to bank existing clinical cell lines in source cells such as fibroblasts and PBMCs. 2, to distribute them broadly, again, to academic and industry worldwide. 3, to make new lines or take them in as appropriate and re-expand. And 4, to edit lines to make isogenic controls where appropriate as well. Next slide. Again, this is just a quick rundown of some of the numbers that we have in our repository right now. So we've got a little over 270 fibroblast lines from a number of diseases within the NINDS mission, also 164 iPSC lines. We have a number of edited lines, some of which are in process as well. If you're interested in those, happy to talk more about that, as well as the protocols in place. I can talk to you offline about how we made those lines. We also have collaborative studies from a number of some nonprofit organizations, but also more importantly, the NINDS Common Fund as well has our regenerative medicine program, including one CGMP line, which industry has been able to access. And then we have active clinical studies, which are contributing their source cell material, either fibroblasts or PBMCs, to the repository for iPSC derivation and editing as well. And then we've distributed, I think almost 1,100 orders we've distributed across the academic and industry. Next slide. Again, this is just the publications that have leveraged Biosend. Again, this is just a small piece of that. We also publish on our repository all the publications that are using those specific cell lines as well. So you can learn more about those and talk to those investigators that utilize those lines. Next slide. Oh, and so that's just me with any questions. I'm going to put my email in the chat. I know we're going to keep moving along. Happy to answer any Q&A in there. And also, please feel free to reach out to me. I'm always happy to talk to any researchers interested in leveraging these supported repositories. Thank you. Thanks, Jane. Back to you.
Thank you, Chris. All right. So yes, that was our first repository. We're going to go through a few more. And then there'll be time for questions at the end. And as Chris said, just put your questions in the chat and we'll get to them. So our next speaker is Dr. Ran Zhang. She's also a program officer at the NINDS. And among her other jobs, she manages the Human Genetics Resource Center at NINDS. Ran, over to you.
Okay, can I have my slides, please? Okay. Here, okay. Thank you, Jen. Good afternoon, everybody, or good morning. So next, I'm going to talk about another NINDS biospecimen repository, which is NINDS Human Genetics Resource Center that is housed at Cornell Institute for Medical Research, located in New Jersey. Next, please. And this resource center is a DNA and a lymphoblastoid cell line repository that was established in 2002. The repository, the mission of the repository is to accelerate discovery of genetic causes and risk factors for neurological disorders by sharing samples and standardized de-identified clinical data. So the repository collects blood samples from individuals with neurological disorders, all from neurologically normal control, these normal control individuals who participate in the NINDS funded studies. And with each sample submission, consistent clinical data also submitted. The data is collected through standardized common data element. So each sample comes with a rich clinical data. This repository not only serves as a central repository, to bank and process samples in a standardized way for NINDS investigators, also serve as a national genetic resource for the scientific community, because all the study participants provided their informed consent for broad sharing of their sample and the data. Next, please. Okay, so currently repository has over samples available from over 42,700 unique individuals. And the bar graph here show you the control as well as disease categories that a repository has sample collected. Also the total unique samples available for each disease category or control. The table on the right is just have breakdowns for the sample types. the LCL cell line, the DNA extracted from whole blood, DNA extracted from a cell line, as well as 96-well DNA panels. And of these numbers, it's just the available unique sample numbers, for each sample type and of each sub-collection. Next, please. Here to show you the NINDS criteria for accepting sample submissions. The studies must be within the mission of NINDS. The disorder of studies, has evidence of a genetic risk, or at least a genetic component. There's a scientific need for the samples in the research community. Usually this scientific need was recognized through workshop, review, or publication. The high quality phenotypic and clinical data are available from the study participants. And again, the study participants, must be consented for broad sample and data sharing. Repository team at Cornell, they provide a site training for sample collection, processing, and shipping. And they also conduct rigorous sample and data quality control and verifications just to ensure, the high quality and uniform samples have been collected. Next, please. This is just the screenshot of the repository catalog. It's show you the information about the collection, the available unique number of samples available for each disease area or control. So if you are interested in learning more about the resource or requesting samples, and this is where you have to start it to start with. Next, please. Here is just to show you, if you don't be bothered to read all this, just want to show you, if you click on the disease on the left side panel of the web page, it will take you to the page, to have the analysis information about the specific disease area, the number of samples available for each group, age group, gender, race, and sample type. Next, please. So to access biospecimens, you need to go to the catalog of a repository. So who is eligible? It's the professional individuals, who are associated with recognized research, medical, educational, or industrial organizations, that engage in health-related research or health deliveries. They can request the samples for their research. And when requesting samples, 2 documents are required. One is a statement of a research intent that describes the proposed research. Another one is a material transfer agreement for biospecimens. So MTA details the terms and the conditions of utilizing the sample. And this document needs to be signed by the sample requester and institutional official who can make a legal commitment, on behalf of the institution. The good part is the sample are free of charge. Even, if you're doing a genomic study, you request thousands of samples, they are all free. But the sample requesters are responsible for shipping charge. Next, please. So this is going to just quickly talk about the sample distribution from the repository. Since their inceptions, over 67,000 samples have been distributed to over 1,000 investigators worldwide, and samples that were banked many years ago, like in early 2000s, continue to be used and cited in the literatures today, that demonstrating their utility. Next, please. You may wondering, what you can use these samples for, and here are examples of the general type of research that these samples have been used for. Detect, identify gene variations, whole genome, whole exome sequencing, gene expression study, functional studies, DNA methylation, and also sample, very often being requested for use as reference materials for assay development. And many of these studies have generated large-scale genomic data, and those data have been shared through the NIH database of genotypes and the phenotypes. We call the DBGAP. Next, please. DBGAP, receives and stores and distributes data for medical research. I'm not going to go to the detail about that data repository, but if you're interested, here is the top is on the left is have the URL and also what is the DBGAP web page looks like. So repository team regularly monitor the new studies from DBGAP and then identify those studies that contain genomic information of the repository samples and then cross-reference these samples with the genomic data at DBGAP. And this cross-reference information also available on the repository catalog. Here are just some examples of DBGAP studies that use the repository samples. Here is with a study accession number, DBGAP study title, number of samples used from the repository, and their genomic assay, type of a genomic assay. Next, please. To date, repository samples have been used in over 800 peer-reviewed publications, and those publications have been cited for over 80,000 times. Next, please. Next, please. Here, I just want next few slides just to show you some high-impact publications that use the repository samples. The previous one is for ALS study, and the Parkinson. Next is a Parkinson study. Next, it's also epilepsy study and the Tourette syndrome studies. I'm not going to go to the details. All these studies, have used many of the repository samples and identified, mutations for the specific disease. I guess I will stop here. Thank you for your attention.
Thank you, Ran. That was great. So now we've heard a little bit more about this program. We are going to hear about the Neurobio Bank from Dr. Abigail Soyombo, who's Program Director at the National Institute of Mental Health. All right, let me get your slides up. There you go.
Great, thank you so much. So hi, everyone, and thank you for attending this presentation where we hope to give you more information about the different resources to support your research. And today I'm talking about the NIH Neurobio Bank, which is a trans-NIH program. I'm one of the co-leads that manages the program. I co-lead it with Dr. Miller from NINDS. He is also present for this presentation. Next slide, please. Great, thank you. So introduction to the Neurobio Bank. It's a consolidated approach to brain banking and a network of 6 brain banks at the universities listed here, situated different parts of the country. These banks existed before the Neurobio Bank was formed. Previously, funded through grants or grant mechanisms, but they were brought together under contract in order for NIH to have more control about tissue distribution. Main goal is to facilitate research, provide access to any qualified researcher, and also to increase brain donation awareness. So the Neurobio Bank has a centralized website where the 6 banks report or deposit data about the specimen inventory. We also have the centralized toxicology testing contract that tests and report on compounds found in the brain tissue. So as I mentioned, it's a trans NIH resource funded by the listed NIH Institute, 5 NIH institutes, and the blueprint for neuroscience research. Under the Neurobiobank model, all qualified funded researchers have access by applying through the website and then submitting your rationale for the experiment. Next slide, please. So the program core activities are below. The outreach to the public to solicit for brain donation. This is mainly coordinated by the Braindono Project. You will hear more about that after my presentation. The sites also have collaborations with different advocacy groups and longitudinal studies. So the brain banks do the brain recoveries, the process, they characterize the subject, post-mortem characterization of subjects, as well as of the specimens, using best practices or rigorous standards. The brain banks also have more than brain tissue, so other nervous system tissues, as well as blood and whatever else that the donors consent to. The sites perform a detailed clinical and neuropathological characterization of the samples, including going through the medical records, accessing whatever information they're able to gain under consent from the donor or next of kin, as well as characterization of the samples. All of this information is provided on the website as well as from the Neurobio Bank sites. So inventory is transparent, is publicly available, and access is managed by NIH program staff. Next slide, please. So the process to request tissue is through the Neurobio Bank website, requirements are that the requester is funded, the institution is registered with NIH, has the federal-wide assurance registration, and researchers also sign an MTA for the distribution. Requests are reviewed by the directors as well as by NIH. We also seek programmatic review. Requests are reviewed as they're received. Final approval is from the NIH program staff, and tissue will be distributed after approval. The process, depending on the scale of your request, would take 8 to 24 weeks. And for much larger samples, it would take longer. Next slide, please. So just to give you some ideas of the metrics, Neurobio Bank processes about 300 requests per year. Next, please, maybe 2 more. Okay, great, thank you. So this shows the age frequencies we have from gestational samples to much older adults, donors. Also showing the race frequencies, we continue to strive for a diverse collection. So this is our current frequencies, but you will hear later from the Braindono Project about the efforts to increase the diversity of our collection. Since inception in 2014, we've processed 2,000 plus tissue requests. Next slide, please. And as I mentioned, the inventory is diverse in terms of race, ethnicity, as well as in terms of the disease categories that are available. Neurodegenerative, developmental, as well as unaffected neurotypical subjects. Next slide, please. Okay, so this just shows some of the recent publications from specimens that were provided by the Neurobio Bank, as well as the number of publications per year. And recently, a new project is the genome sequencing of almost 10,000 subjects from the Neurobio Bank. And the data will be available soon, early next year. So you requesters will also be able to access information about the genetic data from the subjects. Next slide, please. So to contact any questions, you can directly reach out to me or to Olivia or contact us through the Neurobio Bank portal. Next slide, please. And I just wanted to show the network, the Neurobio Bank network. In addition to the 6 Neurobio Bank sites, we have the portal, the centralized, and the testing. A few partners that we work with in terms of sharing best practices and continuing improvement of our methods. And with the Braindono Project, centralized, helping us with the centralized outreach effort. So I'm going to hand off. If you have any questions, please feel free to reach out to me. And this is a list of the trans NIH program officers that we work with. Thank you.
Thank you so much, Abigail. So our next speaker is Tish Hevel, who's the CEO and founder of the Braindono Project, which works very closely with the Neurobio Bank to provide human tissue to researchers. So I will let her take it away.
Thanks, Jane. Sorry. So I'm Tish Hevel with the Braindono Project. It's a nonprofit that supports the NIH's Neurobio Bank, and we serve as their communications and outreach partner. Thought I'd give you a quick look at our job. Let's see. There we go. The assumptions that everybody seems to have about brain donation and how we address them, the mechanics of pre-registration, also what we do in terms of outreach to find potential donors, and then how we engage them. So first, our job. For a little over 6 years now, we have operated in a way that supports the Neurobio Bank. That's our main job. We help people become brain donors by simplifying the process and educating individuals and communities about the critical importance of brain donation. We connect interested parties to the appropriate brain bank for them. So far, about 17,000 people have at least started the process of becoming a brain donor. Seems like the first thing we need to do with a lot of people is correct the 5 false assumptions. First one is that just because you've got the box checked on your driver's license as an organ donor, that includes brain donation. It does not. Separate arrangements need to be made. Or that since you've signed up to be an anatomic body donor, it's the same thing. It isn't. The brain is not used for neuroscience research in that case. A lot of people think only diseased brains are needed. As you all know, unaffected controls are needed in every single study. So we are anxious to reach out to those. It is also not disfiguring, and there's no additional cost to the family to donate through one of the NIH's brain banks. So then we direct interested donors, future donors, to our website at braindonorproject.org. Here's where they learn about brain donation and get their questions answered. When they're ready, they complete a brief online form consisting mainly of contact info, whether the individual is signing up on behalf of themselves or someone else, and whether there's a neurologic diagnosis at play. Once they've completed and submitted the form, then we connect the donor or his or her family to the brain bank that makes the most sense for them. That is based mainly on geography, but there are some exclusionary factors and areas of focus for certain brain banks, and so that comes into play as well. We also let pre-registrants know what to expect when the time comes. And we emphasize that they must talk with their families about their decision. Since the family is the one that notifies the brain bank upon death, they are critical in this whole equation. So once the family member notifies the brain bank arranges transport of the body to someplace local. Typically, the procedure is performed at the family's funeral home these days. The brain bank then sends a pathologist or recovery specialist there to remove the brain and ship it to the brain bank. Also, if the family requests it of the brain bank will supply a copy of the summary of neuropathological findings. That's key information for many families and kind of a motivator in doing it to understand exactly what has gone on. Now to outreach, we learned about this the hard way. That's my dad. He had Lewy bodies dementia. We donated his brain and then formed the Braindono Project to help simplify things. So outreach, we have lots of partnerships with patient advocacy organizations. So that's dozens and dozens of them. We also work with end-of-life professionals, social workers, hospice people, neuroscience nurses. And a big part of what we do is reminding all of these communities that their family and friends can also be donors, even if they don't have a disorder, because that's a great way to support science by donating your brain no matter what's going on. What we do for all these groups is provide customized materials for any method they have of communicating with their stakeholders, all kinds of content, publications, webinars, newsletters, social media, we speak and exhibit, whatever they need from us. We're glad to provide that so that they can help tell their communities how to become brain donors. And we have been very fortunate to land some terrific earned media, they call it. Whoops, don't know what's going on there. Jane, looks like we jumped out for a quick second. Don't know if I did that. There we go. Thank you. Jane, am I messing this up? Tell me if I am.
No, I think that's just the end of the show.
Oh, I'm sorry. There were a couple more slides.
Oh,
so
okay.
just so you know, some terrific earned media is what I was talking about. We were in National Geographic 4 years ago, which continues to produce a lot of pre-registrations. We were on NPR Science Friday earlier this year, which aired on 400 radio stations across the country. So there was a big surge in pre-registrations from that. And then shortly after that, in March of this year, we co-hosted a briefing of the Congressional Neuroscience Caucus. And the co-chair, Earl Blumenauer, put forth a resolution naming May 7th National Brain Donation Awareness Day. So we'll be working hard to get the word out about that again in the coming year. We're also doing a lot of things to focus on inviting all kinds of communities to donate their brains, because as you know, science is not complete unless it is representative of all of us. And I have to end this the way I end every single presentation I do. And that is making sure that you all understand the importance of brain donation. We spend a lot of time thinking about what we're going to do with our stuff and how we're going to, send our, what we're going to do with our money. But your brain may be the most important thing you have to leave behind for a healthier future. So please consider that. Go to braindonorproject.org to start the process. And now I think we're ready for questions. Jane, I'm going to toss it back to you.
So sorry about that. I don't know
No
how
problem.
we lost 2 of your slides.
No problem.
Well, thank you so much for that great presentation and for the very important call for brain donors. So the next portion of this talk, it looks like we have plenty of time for a Q&A session. We're going to have all of the speakers come back and be ready to take your questions. So we'll start with a couple of questions that we received when everyone registered. So the first question is, do you have tissue from the peripheral nervous system that can be used? Any of your resources?
Yes, so the Neurobio Bank, some of the sites do have nervous system tissue and peripheral nervous system tissue. So all of this information is available from the Neurobio Bank website. There's a feature there for non-brain tissue. You can search there. And if you have any questions, you can fill out a form, ask your question. We'll reach out to the directors because some of the inventory is not actually on the website. So we'll find whatever you need.
Thank you so much, Abigail. And I just want to add, if you do have a very niche collection that you need and the Neurobio Bank for some reason isn't able to help, there's another resource that we didn't discuss today that's funded mostly by the Office of Research Infrastructure Program, but also co-funded by the NINDS, called the Human Tissue and Organ Research Resource. And they will help you, I'm just going to post that in the chat. They will help you with your special collections. There is a fee, but the costs are defrayed by the NIH. Okay, let's move on to the next question. What about existing opportunities for comparative biobanks, for instance, in animal models?
So we have our ORIP, which handles a lot of our comparative biomedicine projects. And I'm happy if anybody has questions about detail, I can follow up with you. We have a number of different preclinical modeling biobanks supported through there. I don't know if anybody else wants to add to that.
That sounds great. Thank you, Chris. I put the ORIP link in the chat. So yes, today we're discussing human samples, but there are plenty of NIH resources for comparative medicine. Okay, so for limited resource institutions, are there any recommendations for how to find collaborators to take advantage of some of the repositories that you've talked about today?
I would just ask for at least for the Biosen and NHCDR repositories, we do reach out, we do work with investigators that are in that role. So again, it's kind of one of those questions where I would ask, you can reach out to me and I'm happy to field that with you. I would also mention that if you have limited resources, but say you're just wanting to, let's say, optimize an assay, we can certainly work with you to provide what I call pilot specimens. In that case, it's just a different flavor of a distribution, but we certainly do that on a routine basis. So again, feel free to reach out with me. I'm happy to help assist. Thanks.
Anyone else?
Yeah, I think maybe starting with the NIH, of course, finding, reviewing the publications to identify someone that's working in your area of interest and maybe reaching out to them for collaboration. But while doing that, yes, you can submit a request for pilot study. So you have some preliminary data or to support your research interest.
Yeah, I just want to add, the information about sample submitters, it's not available through the repository catalog. But however, if you're interested in, particular samples and want to seek out more information and get a collaboration with the submitters, you can send a request to repository and they will take it from there.
Great, thank you, everyone. And I think we addressed another one of the questions that was about tissue for pilot studies. So it definitely sounds like everyone, all of the resources are able to provide tissue for doing pilots. All right, so here's another one. Do you need to have project funding to access your resources, existing project funding?
Yes, you do. But if you're applying for, at least for NHCDR and Biosend, you do. But if you're applying for funding and would like to use those resources or plan to do prospective collection, including leveraging those resources, the repositories will work with you and provide a quote and that you can include as a letter of support for your grant application. And that just doesn't even have to be for NIH grant. It could be for other collaborative funding or applications for other institutional funding. So happy to reach out if you have further questions.
Great, great. We have a question in the chat. So what currently exists in the brain bank tissue collection? Maybe Abigail, if you could give in just a brief overview.
Yes, I wanted to respond to that in the chat also. So visit the website. We currently have about 15,000 subjects listed there, diverse disorders and unaffected control, diverse age range, diverse whole brain of sections of regions of the brain. So please visit the website. I'll put the link in the chat.
Great, thank you, Abigail. Okay, all right, so this was also touched on. Someone had asked, do you provide letters of support for grant applications? So I'm seeing a lot of nodding heads. Chris has already mentioned that her resources, they do, and I know that the Neurobio Bank does, and yeah, looks like everyone's nodding. So you'll be able to put those letters of support into your grant application and strengthen it that way. All right, so I'm looking, are there any more questions in the chat that I may have missed? Okay, yes. This is a more specific question, but does the NIH have banked clinical TBI blood samples?
We do. Actually, at BioSun, we recently started distributing samples from our LET-B cohort. That's our late effects of TBI cohort. We have DNA and plasma currently available. There's a few other studies that have just recently started that are natural history studies for TBI. And so those samples aren't available yet, but I would imagine later in the coming year they will be. And if you have an additional questions about how to access those samples or the associated clinical data, again, please feel free to reach out to me and I'm happy to follow up with you.
Jane, if I can jump in real quick, I've gotten a direct message that made me think I should say this to everybody. If you're working with any patient advocacy groups, I don't purport to know all of them, and new ones spring up all the time. So please reach out to me if you want me to connect with a disease community that you are working on. I'm happy to do so.
Thank you, Tish. I think the common thread here is please reach out to us. Everyone wants to help. That's our job. We really want to make sure that these resources are getting into the right hands. It just takes an email. Okay, great. We have another question in the chat. Is there any plan to have specimens from the ventricular walls in the brain? This person studies neural stem cells in the subventricular zone, and it's very difficult to get. Yes, I'm sure it is. So does anyone know about that particular resource?
I haven't seen anything on the horizon for that, but it's certainly within the NINDS mission. So we're happy to explore that. And I can reach out as well to my colleagues who have that vasculature in their portfolios to get additional information. And I'm happy to follow up with you offline, Jashri.
Yeah, and it seemed to me that that might be a project with you probably need to have like freshly isolated, right? Because all our samples are frozen in order to make stem cells. So you probably need to collaborate with what's the HTOR?
Yes.
NDRI, yeah.
Yes.
NDRI, National Disease. I'll put the link also on the side here.
I would just, hi, this is Dan. I would just add, if you're looking for frozen or fixed tissue from the ventricular wall, you can definitely get that from the Neurobio Bank.
Excellent. Okay, I think we've gone through our current list of questions, so I'm just going to give some space for any more questions to come in. I love silence in a meeting. So. Okay, to our speakers, are there any more notes you want to add for everyone?
I just wanted to note that if there are investigators on the call today that are performing research or have previously performed research with existing biospecimens or existing source cells or iPSCs that you think may be of utility if there's sufficient number, maybe it's a rare disease, and something that you may want to help share broadly with the research community, again, please feel free to reach out to us, especially if it's something related to the iPSC, so as we can talk about the source material. And there can be certainly opportunities to bring those existing collections where appropriate into the NINDS research resources. So again, happy to discuss that one-on-one depending on the cases, but I think it's important to note that we do take in existing collections as well where warranted.
Thank you, Chris. We have an additional question that came up. So are there any barriers that program officers often hear about using the repositories, or are there any myths that you would like to debunk here? I'd say the first thing that I had thought when I was doing research is that this would be, highly gated, and that everyone would have to analyze your project first before you were able to get any tissue. But it's really these are supposed to be open resources. They're for all researchers, and all it takes is to ask and reach out to the program officers for that resource to figure out how you can make it work and get a hold of these samples.
Yeah, we've been in the past, when I've connected early on in this process with investigators, there was a lot of questions and misconceptions that it was going to cost a lot of money to pay for access, because some research resources do cost money in a fee for service but BioSun does not. So just note again that you just have to have the funds in place to support that research. We've also heard that again, as you mentioned, Jane, that it sometimes can be an onerous process to access those biospecimens, and maybe it was for biospecimens, 7 or 8 years ago, but we've really streamlined that process. It's not submitting through ERA Commons or anything. We have a very similar streamlined receiving on a rolling basis or applications, a very straightforward review process, and the vast majority of the requests we've approved, things that have not been approved have been, let's say if somebody was requesting cerebral spinal fluid and asking for 2 milliliters from every subject at every time point, something like that. That was, a bit of a pause. We reevaluate the committee and talk to the investigator about, okay, this is a very limited resource, and so we scale back those requests. So again, always room for refinement.
And that's the case with the Neurobio Bank too, right, Abigail? That a lot of times, there are requests that for whatever reason require further exploration. So the Neurobio Bank managers and the actual brain bank directors themselves often engage with investigators to try to figure out what else they can do to help them learn what they need to learn and or specify certain kinds of samples. So there's no dumb questions. Everybody is anxious to see what they can provide to help you advance science. So please keep that in mind.
Absolutely. Thank you. Okay.
And I also wanted to mention, Jane, really quickly, we also distribute to trainees, like many of the biospecimen requests that we fielded and also for NHCDR have been trainees as well. And we just get, co-signatures from their sponsor for those requests. So I want to know, it's not just an established researcher that has access to these. We work as well at all levels of the training for all investigators at each stage in their career.
That's great. I've heard from the Neurobio Bank directors that they have various levels of people coming at various knowledge levels of people that come in with requests, and they are happy to sit down, virtually sit down with you and discuss what kind of brain tissue you actually need for your project, because they're experts and they know a lot about brain tissue. So they're great resources. Okay, thank you, everyone. I don't see anything else in the chat. Great. Then thank you all so much for joining us. Anna, would you like to take over?
Yes, I just wanted to conclude this webinar and say a huge thank you to our panelists. A big reason that we started this series and we are hosting this webinar is to put a face to a name or put a face to a repository. So there are people behind NINDS and these repositories that are a huge resource for you. Sometimes you don't know what you don't know what to ask for. So hopefully this webinar is a great resource. Please tell your colleagues about it. We'll share the recording, we'll share everybody's emails, we'll share all of the links and resources and PowerPoint presentations. So please forward that on to your colleagues, let them know about all these wonderful resources that we have for you. So with that, I just want to say thank you again for attending. We'll be in touch very soon, probably next week, with all these resources. And please join us for our next Open Stage webinar on January 30th. That will be on biomarker development and validation, translating discoveries into fit for purpose clinical tools. I'll post the link to the chat to that event there. So thank you so much again, and hope you have a wonderful holiday season ahead. Bye.
Thank you.